Okay, so for chapter 26, there's not much that needs kind of further explanation or time to go over a lot of the details. More of them are just kind of stuff where it's just basic stuff to learn. There's not much explanation to go behind it. So I'm going to go back to towards that type of highlight type of situation where I just kind of pick through certain points of the PowerPoint to explain. And so this video is more like a highlight for chapter 26. So chapter 26 is all about nutrition and taking in of our different nutrients coming from the foods we eat. So to regulate whether or not we are signaled to eat food or not, that's regulated by different hormones. So here at the bottom, you have your ghrelin, your PYY, and your cholecystokinin. All three of these are short-term regulators of hunger, so regulating hormones for regulating your hunger sensations closely around meal times. So in our little chart, the green arrows are for having a stimulatory effect when they bind to neural networks in the hypothalamus in the brain. And then red arrows show a inhibitory effect when binding to those neural networks in the hypothalamus. So both your ghrelin has a stimulatory effect in the hypothalamus on your neuropeptide Y secreting hormones. So this causes the release of neuropeptide Y that causes your increase in your hunger level. So when your stomach is empty, ghrelin is secreted to get you to basically feed yourself. So here we have our little piece of cake as in stimulating the want to eat. PYY and cholecystokinin are released later on in the process when you've eaten and you're starting the whole digestion process. So they're released and they have a inhibitory effect, so still binding to those same neuropeptide Y secreting neurons and therefore inhibiting the hungry signal. Your other two hormones that affect your hunger sensation are insulin and leptin. Both of these work long-term on your hunger sensation. So insulin, both of these also tell your brain of how much fat you have. So insulin also is inhibitory to your neuropeptide Y secreting neurons, so inhibiting that hunger sensation that's caused by the neuropeptide Y. For leptin, secreted by your adipocytes, so your fat cells. So that tells your brain that you have enough fat, depending on the levels of leptin. So it has a stimulatory effect to a different type, different neural network in the hypothalamus, which are your melanocortin secreting neurons. And when those neurons secrete melanocortin, then it tells your brain that you're had enough to eat, and so you have more of a satiety or feeling full and not hungry effect. So here we have our little picture where the person is saying no to the ice cream for whatever reason possible. Who says no to ice cream? Okay, so when our body tells us that we are hungry and we take in food, part of what we take in have different nutrients in them. So there's 6 classes of nutrients. You have water, carbohydrates, lipids, proteins, vitamins, and minerals. So your water and your carbohydrates, lipids, and proteins are called macronutrients because you need large quantities of them. So thinking of the size of how much food you eat in a day compared to your vitamin and mineral needs, which can be satisfied by a single, multivitamin that you can take in the morning with a meal. So that's a micronutrient because you only need it in a small amount. So skipping ahead to lipids. So because lipids are hydrophobic, which means they do not like water and therefore will not dissolve in your blood plasma, they have to be transported into their own vehicles. And those vehicles are called lipoprotein complexes. So there's multiple types that we'll go through in a little bit. So the lipids are coated with proteins and phospholipids as a package that allows the lipids to be suspended in the blood so that it can be transported in the bloodstream. These lipoprotein complexes excuse me, also serve as a recognition marker that tells the cells what's in that package, and therefore the cells can then absorb the lipids from those packages. Oops. So your blood serum lipoproteins have 4 major categories, and they're based by how dense their lipid contents are. So here on the side in our little coating for our little pictures, the little orange is your triglycerides, the brown is cholesterol, the blue glob here is a protein, and then these little here on the outside are your phospholipids. So your first type are chylomicrons. So earlier when we talked about, I believe it was chapter 25, once your lipids are digested and then they're absorbed in your small intestines, and then as they leave, these small intestine cells are packaged into these chylomicrons so they can be taken up by the lymphatic vessels to be transported to the blood. So in those packages or those chylomicrons, there's actually very little triglycerides. Then a little bit denser than that and also containing more protein is a very low density lipoprotein. So again, having a little bit more triglycerides than your chylomicron. Even more dense is a low density lipoprotein. So here in the orange is full of just straight lipids, and then there's a cholesterol core. And again, more protein on this one as well. And then a high density lipoprotein that has a whole lot less cholesterol and way more fat. So higher density of fat compared to the cholesterol. And again, more proteins on that package as well. So with these different packages or vehicles to transport fat, not only are they different in their composition or what makes them up, they also differ in their function. So kind of review what I just said earlier as well as what was covered in chapter 25. So lipids are absorbed in the small intestines. They're packaged into chylomicrons where they enter your lymphatic vessels, traveling through the lymphatic system to be dumped into the subclavian vein, into your circulatory system. And when they are absorbed by the blood capillary cells, there is an enzyme called lipoprotein lipase that hydrolyzes or breaks apart the triglycerides into monoglycerides and free fatty acids. So it's taking off those free fatty acid chains that are on the outside of that triglyceride molecule. Then those monoglycerides and free fatty acids are transported to your adipocytes, and that's where they're repackaged as triglycerides. So the chylomicron, after it's gotten rid of its triglycerides, then that triglyceride is then sent to the liver so it can be degraded. So now moving on to the different functions of your different lipid packages. So your VLDL, or your very low density lipoprotein, produced by the liver, and its job is to transport the lipids to adipose tissue so they can be stored. So once those triglycerides are removed, you still are left with the cholesterol core. So it goes from being a very low density lipoprotein to a low density lipoprotein. So low density, so it becomes a low density lipoprotein. These are also can be produced by the liver. They are mostly a package for cholesterol. And then once those LDLs circulate through the body, they can be absorbed by cells that need cholesterol. And then the package, sorry, not that. Once the LDLs are absorbed into the cells, so they go through endocytosis where they kind of wrap around the LDL and bring it into the cell, then lysosomal enzymes will release the cholesterol, and then the rest of the LDL package is broken apart. For your HDL, it starts off as an empty package, still being made in the liver, but its job is to circulate throughout your bloodstream to pick up cholesterol and phospholipids from other organs. So basically, instead of transporting lipids to get to a location, they're going to different locations to pick up lipids. So it acts as a vehicle to remove excess cholesterol, helps to maintain a total plasma concentration of less than 200 milligrams per deciliter. So it takes those cholesterol and brings it back to your liver. So with the cholesterol in your body, actually most of it can be made by the body, so internally synthesized, so made by the body, rather than being taken into by a dietary source. So depending on how much you take in through your diet, then your body will compensate for the lack or excess of cholesterol. So if you have a high dietary intake, then the liver will lower its cholesterol production. If you have a low dietary intake, your liver will raise production. So it still helps to maintain your cholesterol levels. So with two of those packages, your LDL and your HDL, the levels of them is a determinant of your levels of health. So high levels of LDL is a warning sign that there's cholesterol deposition in your arteries, since you have these packages that are full of cholesterol being circulating throughout your bloodstream. With your HDL, so again, your vessel package that travels around to pick up that excess cholesterol as well as phospholipids from the different cells and then taken to the liver to be disposed. That actually is very beneficial to the body, so it helps to maintain the lower levels of cholesterol and keeps it from clogging up your arteries. So what's recommended is to increase your ratio of HDL to LDL through using regular aerobic exercise, as well as avoiding smoking, saturated fats, which come from your animal fat sources, as well as coffee and stress. And I put my little laughing smiley face as in, yeah, that's going to work for students who are, stressed out and having to work and balance life and all that stuff at the same time. So I'm going to move on to our next thing. So moving on to using those nutrients and how we get the energy from them. So starting with carbohydrates. So most of our dietary carbs are used as a fuel within hours of absorption. So in order to use up our carbohydrates as fuel, the carbohydrates have to go through what's called glucose catabolism. So when those carbohydrates are digested, broken down, eventually becoming glucose, that glucose, which is this molecule here, when you add glucose plus oxygen, you end up with carbon dioxide and water as a byproduct, and we'll go through that whole process later. But basically, the function of this is to transfer the energy from glucose to your ATP so that those ATP molecules can be used as an instant source of energy. So the definition of glucose catabolism, so here's the bullet point version, and here's the everything all together in one sentence version. So in glucose catabolisms, there's small steps throughout this whole process that are controlled by separate enzymes. And through the actions of those enzymes, they release energy in small amounts. Those small amounts of energy are then transferred to ATP. This isn't a perfect process, so some of the energy that is released as heat versus being transferred to ATP, but that still helps our body maintain its core temperature. So there's three major pathways of glucose catabolism. So you start off with your glucose molecule. That glucose molecule will first go through glycolysis. And during this process, it will split into two pyruvate molecules, making two ATP molecules. And we'll go through this later. The next pathway is anaerobic fermentation. So this occurs in the absence of oxygen or low oxygen levels. So it takes these pyruvate molecules from glycolysis and it reduces them to lactate. So if something is reduced, it receives electrons from another molecule. So that reduction process, in a sense, ferments the pyruvate to lactate. The last pathway, and this one only produces two ATP molecules. Your last pathway, which is way more efficient, is aerobic respiration. So this occurs in the mitochondria, occurs in the presence of oxygen. So when there's high enough oxygen levels, your body will continue with aerobic respiration. And in aerobic respiration, the pyruvate is oxidized, so it has its electrons stripped off of it. That pyruvate molecule then eventually becomes carbon dioxide, which is exhaled, as well as H2O, which the body just uses. So it's your body's own way of making its own water, and that's called metabolic water. So it's one of your water sources, which you'll cover later in the urinary system. Actually, it's the next module, how your fluid is used throughout the body, and so metabolic water is one of your sources. So during these process of glucose catabolism, different enzymes will remove electrons in the form of hydrogen atoms. So the proton from the hydrogen atom is removed, but the electrons are also removed. Then they transfer those hydrogen ions to different coenzymes, so pass it off to the next player. Those coenzymes then pass those hydrogen atoms off to other compounds, where they help with other pathways. So your hydrogen atoms are split into the protons and then to the electrons to have other functions for making ATP. So even though you have all these enzymes to do the work for glucose catabolism, you need those coenzymes to transfer the hydrogens to the different enzymes. So the two coenzymes that's very important for this situation are NAD+, which will bind to two hydrogen atoms. So when hydrogen is stripped off of a molecule, it's done so in pairs. So with NAD+, it binds to one hydrogen atom as well as one electron from the other hydrogen atom. And then there's this free proton that's left over. For FAD, it binds to both, two hydrogens still again, but it binds to both the protons and the electrons from that hydrogen. So these are basically carriers to the next enzyme. So like I just said, hydrogen atoms are removed in pairs, so two protons and two electrons, and then they're transferred to the enzymes, which increases their free energy for that enzyme, and then they become the temporary carriers of the enzyme to their next step. So I just mentioned that earlier, binding to your electrons as well as the protons for FAD, and then binding to only one of the protons for NAD+. So this chart has up to here all your steps for glycolysis. You might actually have to have all those steps for another class. I will go over it very lightly because you don't need all that information for this course, but still kind of important to at least kind of go through it in a kind of general sense. And then maybe you have to do it later in more detail, but at least you'll have the kind of very basics first, and then you can build on it in a later class if you need to. So starting up here, glucose is a 6-carbon molecule. So each of these little gray balls here is a carbon molecule. Then going throughout that whole process, that one glucose molecule is converted finally down to two pyruvate molecules. Pyruvate is a 3-carbon molecule, so the 3 balls here representing there. And then later, those 2 pyruvate molecules will either go through anaerobic fermentation or aerobic respiration depending on your oxygen level. So low oxygen, anaerobic fermentation. Plenty of oxygen, aerobic respiration. So aerobic respiration also includes your citric acid cycle as well as the electron transport chain. So here in our little figure are 2 pyruvate. So here we have low oxygen, so going through anaerobic fermentation where that pyruvate molecule is fermented to lactate. And in this process, the NADH molecule is regenerated so that it can go through glycolysis again and therefore making 2 more ATP. For aerobic respiration, there's a whole bunch of steps. It has its own little figure, and we'll go through a bunch of those, go through those steps later. So I'm going to quickly go through glycolysis. There's multiple steps. There's a phosphorylation step that uses up 1 ATP. It also helps to prevent sugar from leaving the cell. So this glucose 6-phosphate molecule, so having that phosphate group on there, what that does is it changes the molecule enough that prevents glucose from becoming highly concentrated in the cell so you can continue bringing glucose into your cell. That glucose 6 molecule can also be used to help store glucose in the form of glycogen or made into fat. There's other steps that happen. You have a priming step, so another phosphorylation using up an ATP. Then this molecule is broken up in 2 different ones. So here we have the kind of precursor to our 2 pyruvate molecules. And then finally, those 2 molecules are phosphorylated. So breaking off, not phosphorylated, dephosphorylated. So those phosphate groups are taken off that molecule and then transferred to an ADP, making them ATP, and that's where you get your making of your 2 ATP molecules. So here as well as here. So technically you're making 4, but because you use up 2 ATP molecules during this process, you only make 2 net ATPs out of that glycolysis process. Okay, so here's in text. So although 4 ATP molecules are produced, the net gain is only 2 ATP molecules because you use 2 during the process of glycolysis. So with glycolysis, some of the energy is in the ATP that's created, and some is in the NADH molecule, some is lost as heat. Most is still stuck in the pyruvate molecule, so that's why you can continue using that molecule to either go through anaerobic fermentation in low oxygen or aerobic respiration in high oxygen. So the end products of glycolysis are your 2 pyruvate molecules, your 2 NADH coenzymes, and 2 ATP molecules, as well as the 2 hydrogen protons, so the ones that didn't bind to the NADH molecule. So next, I'm going to make another kind of second set for this video to go through the anaerobic fermentation as well as the aerobic respiration and maybe some other stuff. Just, again, looking at the time for the video.